then. Well, thank you. See you later. Ni hao. And Professor Wang, thank you very much for that lovely introduction. Xie xie. I've chosen this morning to talk to you about the way in which physiology can be reintegrated with one of the mainstreams of biology, that is the theory of evolution, because I believe that one of the major changes in the conceptual foundations of biology are occurring in the field of evolutionary biology. Moreover, those changes are occurring in a direction that will enable physiology to become more relevant. Just a little bit of brief history, though. If you go back about 200 years to Jean-Baptiste Lamarck, the French biologist who published the Zoologie philosophique, he established the transformation of species. It's also well known that he assumed in his work that there could be the inheritance of acquired characteristics, and for that reason that process is sometimes called Lamarckism. But he didn't invent the idea. He assumed it, as others did too, from time immemorial, and in particular Charles Darwin, who 50 years later published The Origin of Species and proposed the theory of natural selection. He also assumed the existence of inheritance of acquired characteristics. The significance of this will become later in the lecture. And then around 1900, there was the integration of Mendelian inheritance, that is discrete inheritance, with evolutionary theory. And about the same time too, Weizmann established what was called the Weizmann barrier, the idea that the germ cells and their genetic material is not in any way influenced by the organism itself or by the environment. And then, something like 40 years later, a variety of people, Julian Huxley, R.A. Fisher, Haldane and Wright, put things together to call it the modern synthesis. So what exactly is the modern synthesis? It's sometimes called Neo-Darwinism, and it was popularized in the book by Richard Dawkins, The Selfish Gene, in 1976. Its main assumptions are, first of all, that it is a gene-centered view of natural selection. The process of evolution, therefore, can be characterized entirely by what is happening to the genome. It would be a process in which there would be accumulation of random mutations followed by selection. Now, important point to make here is if that process is genuinely random, then there is nothing that physiology, there's nothing that people like you and me can say about that process. That's a very important point. The second aspect of neo-Darwinism was the impossibility of the inheritance of acquired characteristics. It was miscalled Lamarckism. As I said earlier on, Lamarck did not invent the idea, he assumed it. And there is a very important distinction, particularly in Dawkins' book, The Selfish Gene, between the replicator, that is the genes, and the vehicle that carries the replicator, that is the organism or phenotype. And of course that idea was not only buttressed and supported by the Weissman barrier idea, but also later on by the central dogma of molecular biology. All of these rules have been broken, and that is the subject of my lecture. First of all, are mutations random? A very important book to catch up with what is happening very rapidly in the field of evolutionary biology is the book by the Chicago biochemist James Shapiro, Evolution: A View from the 21st Century. He writes and he gives detailed evidence. He has thousands of references on this in his website. It is difficult, if not impossible, to find a genome change operator that is truly random in its action within the DNA of the cell. All careful studies of mutagenesis find statistically significant non-random patterns of change. In other words, there are hotspots in the genome. Moreover, as we will see later on, the frequency with which those changes can occur can respond to what the organism is doing and what its environment is doing. Just to give one example, p-element homing in fruit flies, these are DNA transposons that can insert into the genome in a functionally significant way. And what has been shown is that they do so with a frequency that is 50% greater in regions of the genome that are related functionally. We don't at the moment understand that, but one possibility is that it depends upon the organization of the genome, the way the DNA is folded and the way in which it is structured around its proteins. Moreover, not only is it the case that mutations are not random, another major assumption of the modern synthesis is incorrect. And we found that out at the time of the sequencing of the human genome in the year 2000, because the Nature report of that sequencing showed that two major groups of proteins, the transcription factors and the chromatin binding proteins, do not show gradual change between species of the sequence of the proteins, but major domains switching into one protein after another. This is from the report, the 2001 report of the sequencing of the genome, showing domain accretion in yeast, in a worm, a fly, vertebrates, and human. And the stars indicate, that's all you need to take home from this slide, the domains that have shifted around as whole domains, not gradual mutation, one amino acid after another. And as you can see, there are many of them. The same is true for transcription proteins. So my first conclusion is this. Not only is mutation not random, that was one of the essential assumptions of the neo-Darwinian synthesis, but proteins, at least some of them, did not evolve via gradual accumulation of mutations. So let's return to the popularization of neo-Darwinism, which is the book The Selfish Gene. Is it metaphor or is it empirical science? In reply to a philosopher, Mary Mitchley, who wrote a review, a very critical review of The Selfish Gene back in 1979, remember the book was published in 1976, he replied to Mary Mitchley that it wasn't a metaphor at all. I believe it is the literal truth, he wrote, provided certain key words are defined in the particular way favoured by biologists. Now, if you haven't understood that statement, I want to teach you what a metaphor is. A metaphor is precisely key words defined in particular ways where you change the meaning of the word. What happened there is that, I'm sorry to say, that a very distinguished scientist, because Richard certainly is that, and a very, very good writer too, didn't understand what a metaphor actually meant. For those of you who want to follow up these philosophical and linguistic points in greater detail, the article that I published last year in the Journal of Physiology goes through this point in much more detail than I can do in a general lecture. The point is very simple. A metaphor does not cease to be a metaphor simply because one defines a word to mean something other than its normal meaning. Indeed, it's the function of metaphor to do precisely this. So let's do a little experiment. This is an experiment on you. I'm going to compare the two possible metaphors here, genes as prisoners versus selfish genes, or genes as cooperators, if you like, rather than selfish genes. It doesn't really matter. The question I'm going to put to you, as I put two texts up, is what conceivable biological experiment could distinguish between the two groups of metaphors. So let's take the selfish gene metaphor. He wrote, it's beautiful language, I have to say. Richard has an ability to write that is admirable. Now they, genes, swarm in huge colonies, safe inside gigantic lumbering robots, that's you and me, of course, sealed off from the outside world. That is the Weissman barrier, that is the central dogma of molecular biology. Communicating with it by tortuous indirect routes, manipulating it by remote control. That's the gene-centric view. They are in you and me. That's the only statement that is empirical. They created us body and mind, and their preservation is the ultimate rationale for our existence. And in case you didn't understand the selfish gene, he went on to write...